We've gotten away from bacteria now. We talked about polycistronic DNA, we talked about operons, we talked about how bacteria can produce only those materials they need when they need them in response to the environment. Well, eukaryotes need to do the same thing. They don't want to have excess protein product. They want to only have what they need when they need it. But because of the divergence in evolutionary pathways, eukaryotes have slightly different ways, very different ways of regulating their genetic transcription, of making sure they've only got the right protein product when they need it. And that's what we're about to cover.
But we're not bacteria, we're eukaryotes. We got a little bit more complex system, and I know you cringe when I say that. I'm not going to go into all of the vast complexity of how eukaryotes control their genes. The big thing to note is that eukaryotes control their genes using what are known as transcription factors. Back to milk. You have two kinds of transcription factors control the rate of transcription. You can control it through general transcription factors, GTF, or regulatory transcription factors. In the end, ultimate big picture, transcription factors are going to speed up or slow down transcription by helping the RNA polymerase either bind or by stopping it from binding. So these are factors that bind to the RNA polymerase, and they help it hold on to the promoter. That's it. Regulatory transcription factors are going to give it either a push or hold it back. They're sort of like literally molecules that will sit behind the polymerase and push it, or molecules that will chain it to a certain area, slowing it down. Have you guys ever had kids like holding onto your legs while you walked? Yeah, so that's, sorry. That's what a regulatory transcription factor would be like. It would slow it down. Yeah, it's true. And again, we've got activators and repressors. What does an activator do? What does a repressor do? Any questions?
Is there a way to get a child that will speed you up?
Depends, I'd say. How cold are their hands?
Good point.
So these activators are going to bind to regions called enhancers. Not terribly important to know the difference there. And then the repressor is going to bind to areas called silencers. These are just enhancers and silencers are regions of DNA. No, the big thing is activator, repressor. And I think activator, repressor is not bad for you guys because literally they say what they do. They activate things or repress them. Enhancement is a region of the DNA.
The activator binds to the
Enhancer, yeah.
And the repressor
Binds to the silencer. Sure. Now, most of these genes aren't just regulated by one thing or another. They're regulated by a whole bunch of different things. And that's why you get, like we said earlier, so many different levels of expression. So many in-betweens. You might have one or more activators working on the same polymerase. You might have two activators and a repressor working at the same time. So you're not always producing at top speed, and you're not always producing at lowest speed. You've got lots and lots of space in the between. Yes? The Tata box is the DNA, yeah. This is, so, not necessary for you guys to know, but the bonds holding adenine and thymine together are the weakest of the binding. So that's where helicase will bind. It's the easiest place to open. So whenever you see an origin of replication, we'll have a lot of A's and T's.
So I get the C's and the A's then the R's
So CA and R, those are not, that's not cytosine and adenine.
Oh, okay.
Those are just proteins, and I'm not going to ask you to know those. What I'm saying, the reason I put this picture up is because you can see lots and lots and lots of transcription factors impacting one polymerase.
Myocardium, is that
Yes. I think I see where you're going with it. They might also change the composition of the nucleosome. Do you guys know what a nucleosome is? Have we talked about that at all? No. Sorry. A nucleosome is a piece of DNA wrapped around a histone. What's a histone? A histone is what histone is a protein that helps to take the DNA and condense it. So you guys, back to my shoe. All right, so you got DNA. DNA is a double helix, right? In order for DNA to be compressed from chromatin to a chromosome, you need to get it super, super highly condensed. It starts bunching up. And what it bunches around, like, ow, that hurt.
Like all the way up.
Yeah, when it bunches like that, my finger would be considered the histone. Okay? So it provides sort of a nucleus for it to wrap around.
So the nucleosome is what the histone wraps around.
Right. No. The nucleosome is the histone and DNA. It's that unit.
It's the center of the DNA.
Yes, you can look at it that way. You may inhibit DNA from being read through methylation. Do you guys remember? Methylation turns DNA off. You know the X chromosome. Let's take the X chromosome. That gets methylated. It gets turned off. So that other polymerase can't bind to it. Methylation turns it off. And if you've turned off the gene, what happens? You don't get any gene product. The reaction rates, whatever it may be, go down. You might have upregulation. An enhancer causes upregulation, speeds it up. Or you may have downregulation, slows it down. Do you guys see a theme working out here? Activators speed things up, repressors slow things down. You get upregulation, you speed it up using an activator. You get downregulation, you slow it down using a repressor. These are the big picture things I want you to take away. Ups and downs. Typically, what you're regulating is really, really close to the promoter. Why do you think activators and repressors are always so close to the promoter? Not trying to trick you. Right, what has to land at the promoter? The polymerase. So by being near the promoter, the regulatory elements can directly act on the polymerase. Nope. I have a lot about activators and repressors. Right. It is. Do you guys have any questions? I'm not going to ask you specifics about transcription factor 2D because I think, truthfully, you may understand how it works. If I've got an activator near transcription factor 2D, hypothetically, what do you think is going to happen to the gene that it controls? Speeds up, right. And if I've got a repressor near it, slows down. Sometimes you're going to have a mediator interact with the activator and the polymerase. A mediator is a nice little cushion. You guys have probably acted like a mediator in your house at some point or another. Yeah, and you're like, yeah, that's sort of my job. It just makes things move a little bit smoother.
Transcription in eukaryotes can be complex. It involves not just making sure that you can get a polymerase to the right place at the right time. That's ultimately what it is. Make sure you can get a polymerase to the right place at the right time, and that polymerase is released to do something. But it also involves the structure of the DNA. You might have your DNA tightly bound around histones and have only one area open. But then that histone can shift down, and that opens up a whole new area of DNA for the polymerase to bind to. You've got repressors and activators. Activators speed up transcription by helping a polymerase bind, an RNA polymerase bind. Repressors slow it down by stopping that polymerase from binding. And then you've got all of these transcription factors, general transcription factors that may contribute to helping RNA polymerase bind or stopping it from binding. There's a lot going on there. MicroRNAs, non-coding RNAs, all of these can be involved in transcription and translation of eukaryotic genes. So we're away from operons, but we kind of jumped into a frying pan with slightly more complex and interconnected molecular mechanisms. Regardless, using transcription factors to recruit or stop polymerase from binding, you can the eukaryotic cell will be able to respond to its environment with genes that it needs when it needs them. These content review questions are here to focus your studying. You focus on this particular lecture. The next mini lecture is going to be about the impact of non-coding RNA, because apparently there's a whole bunch of RNA out there. It's not just mRNA, tRNA, and rRNA, but there's a whole bunch of other RNA that's involved, especially in eukaryotic gene regulation. So that's coming up in the next mini lecture.